Hi, my name is Cole Pearson. I work with Dr. Joseph Munyaneza out of Wapato, Washington, and also collaborate with Dr. Lane Backus in Parlier, California. And I work on psyllid feeding behavior studies. And what we do there is we actually use a machine that allows us to monitor their feeding behaviors while they're actually probing inside the plant. And that allows us to see what kind of tissues they are in and what behaviors they're exhibiting during their feeding behavior, because normally you can't see that with them just sitting on the plant, because they insert their stylets, they go down through the tissue, and then you don't really know what's going on. We figured out a way to quantify that, and what we've been able to do thus far is identify what we call waveforms that represent different behaviors, and those are going to be used to test and look for host plant resistance. So there's one waveform in particular we're really interested about. That's the ingestion from the phloem. So if we really don't see that and we allow it to feed on a certain cultivar for quite a while and we don't ever see it make it down the phloem, well, the pathogen itself, Larobacter, is phloem limited. So if the insect's not able to get the phloem, well, then it's not able to actually inoculate the plant with the bacterium. And we're also want to use these for pesticide trial studies to test the effectiveness of pesticides. Also look at the systemic effects and how long they actually really last, because this will give you a good idea, because you can see exactly when the insect starts feeding and exactly when it stops, and it gives you the length and the duration of which it actually is feeding or not feeding, and that'll allow us to give us some good hints and maximize the effectiveness of pesticides used in pest management regimes.